So today we're going to go over blotting techniques. I'm going to go over the Western blot, the Northern blot, and the Southern blot. And blotting techniques are basically techniques that allow for detection of specific molecules. So, big examples of these molecules could be DNA, that could be a big one. Proteins is another big one, and RNA is another big one. And these are the three we're going to be studying today, beginning with the Western blot. And as always, I'm going to start with a motivation. Let's imagine we have our killer T cells, right? Or let's say natural killer cells instead. If you don't know what these are, just know they're immune cells that like to kill cancer cells, all right? And these cells have something called granzymes. I'll put them in red, right? And let's say we want to figure out how many granzymes are in one of the natural killer cells and another cell. Let's say an inactive natural killer T cell, right? This for the purposes of studying immunology. This guy's sleeping. He's sleeping, and he'll probably have less granzymes, right? Maybe not nearly as many. And we want to determine the relative abundance of these two granzymes. So how can we do that? Well, let's see. We can lyse this guy, right? We can break him apart, and now we can get a bunch of these granzymes, and we can lyse this guy to get a few, right? But what can we actually do with this? Now we only have, one of them has a lot, the other one has a little, but we don't really know how to be able to figure out how many of these granzymes are, right? And that's where we can use our blotting technique, and that's where we use Western blot. And Western blot, what we basically have are antibodies, right? And what antibodies are basically just these little things, these little proteins. They look like this. With like disulfide bonds there, okay, and basically they can bind to a protein or antigen or something, right? And these antibodies, we know what they can do. We know the protein they bind on, and a Western blot basically determines the presence of a protein. That's basically all it does, and we'll see exactly how it does that. But the idea is that we'll have a protein, and then we can use a specific antigen and basically mark that protein so we can see it, right? And going back to the relative abundance, if we have a lot, let's say we have like a dye on this antibody, right? If it's a lot brighter, then that means we can safely assume there's a lot more of it compared to a more dimly lit one. I'll explain more of what that means in detail. Let's go over the procedure first. First, we start off with SDS page. So imagine we have a little vial of proteins, right? A bunch of proteins in here. Watch my video on SDS page. Basically, what that does is we end up with a little gel with proteins sorted by, this is an SDS page, by size only, right? So we might have bigger proteins near to the top and smaller proteins close to the bottom. And maybe we can show that here, right? And basically what we do with this gel now, so we took this gel out, and we want to place the gel on top of a membrane, okay? So let's show this membrane in green. All right, let's say we place it on this membrane, this green membrane right there. And what this membrane is going to do is basically act as a little anchor for our proteins. Oops. And so basically, once we have this membrane here, what we do is we actually sandwich this guy with filter paper. I'll do the filter paper in yellow, right? Filter paper is not very important in terms of what we need. What we need is actually the buffer and an electric current using a positive cathode and a negative anode, right? A negative cathode and a positive anode. So we have this filter paper, and then we have our cathode. Let's put it here, and we have our anode. And then we run this guy in some buffer, and what happens when we turn this on is that the negative proteins, right, since they're covered with SCS, will migrate downwards, right, onto the membrane. And we do this for a while, and then once we believe all the proteins are on the membrane, we take off the gel, right? And what we do is we block it. We can block it with something like milk or BSA, and all this does is prevent antibodies from binding to the membrane. Remember, So now we have our membrane. Let's do it in green. And this membrane is pretty thin, right? Not too thick. And there's a bunch of proteins on it now, right? We just got basically an exact replica of what we had before on our gel, right? And we put milk on it so that antibodies, right, won't bind to the membrane. You can imagine how that could be a problem. If our antibody is going to bind to this protein, we, in order to detect it, right, if the antibodies end up binding to the membrane, then we're kind of, there's kind of no point, right? But so we block it with some milk, so now antibodies can't bind to the membrane. Now we have something called a primary antibody, and now the primary antibody will bind to our protein of interest. Let's say this is our protein of interest right there, and this antibody will bind to it. All right, let's say there'll be a bunch of antibodies, right? And then we will wash it. We'll wash this guy, and so all that's left are the antibodies on this protein, right? And that's our primary protein. Then we have a secondary protein, and with the secondary antibody, what the secondary antibody does, let's do it in purple. This guy will bind to the primary antibody, and this guy usually has some sort of dye that can be detected. All right, this guy has a dye that can be detected, and then we wash it again, right? Now, what is this washing exactly? Well, when we first put the primary antibody on, right? When we first put the primary antibody on, there's going to be a bunch of these guys just like scattered across, right? But the only ones that matter are the ones bound to the red protein, right? And so when we wash it, all these guys will go away aside from the original. And same with the secondary antibody. And the secondary antibody will have some dye. Let's say it's a fluorescent dye that can absorb UV light and then, disperses it afterwards. We can just not detect this protein, right? And if we see any detection, then that's great. Then we found the protein of interest. And if there's no detection, right, if there's no dye, if there's no light, then chances are the protein wasn't there. And we can take a look at a quick result. We can see that this is a lysed cell, right? And we can clearly see that it has some sort of protein that was able to be identified by the Western blot. And we can see there's a lot more here with this many micrograms versus that many micrograms, right? Which should make sense, right? And that's basically what the Western blot. Basically, we get to figure out if a protein is in a sample. Now let's go to the Southern blot. And the Southern blot is a little different, and this concerns DNA. Okay, so we'll start with our motivation. Imagine we have a big genome, right? Let's say we have a cell here, and this is a huge cell with a bunch of genes, right? Let's say we want to figure out if this cell has a specific gene. Let's say gene A, right? And how can we do this exactly? Well, if you think about it, probably the best way would be to denature these DNA and then have like a probe to get in there so we can see it, right? So that's the idea. We have our gene of interest, right, and we want to bind it to a probe with, let's say, like a fluorescent marker so we can see it. But how do we do this exactly? I mean, this makes sense on paper, but how do we actually go about executing this? Well, and that's where the Southern blot comes in. Like I said, it uses DNA probes. So what is a DNA probe exactly? It's basically a single strand of DNA with some base pairs and a fluorescent dye, or some sort of indicator. It could also be radio labeled phosphate backbones, right? So that we can detect the different isotope later on through like UV vis, right? But either way, this DNA probe, what this will do, it will bind to our gene of interest or our sequence of interest. And that's sort of the idea. And this will be able to detect specific DNA sequences because we can synthesize these probes to be whatever we want, right? And let's take a look at the procedure. First, we have whatever, the genes we have, and we treat it with a restriction enzyme digest. And what this basically does, we do this upstream and downstream of the gene we really care about, and basically this just cuts our genome into a bunch of small pieces, right? So that now we have just like varying strands of DNA. And we know, and since this digest is upstream and downstream of a gene, then we will have guaranteed that our gene wasn't like cut in half, for example. Now, what we do with all of these, right? Let's say we put it into like a little vial, is we run it with agarose gel electrophoresis, right? And then we also treat it with NaOH, sodium hydroxide. And this will basically just pull apart these DNA to make them single-stranded. So what we're left with, if you're unfamiliar with agarose gel electrophoresis, watch my video on electrophoresis. And basically this does is separate these DNA based on size, right? So we have like varying sizes here, right? And now we do some stuff that's very important. We have our little buffer container here, right? Let me draw my buffer container, right? And it's filled with like buffer in here, right? It's filled with buffer. What we do is we first add our filter paper, right? Okay, this might be hard to draw in 3D, but imagine we have our filter paper, and this filter paper is wet. All right, this is wet paper, filter paper, wet. And then we put our gel onto it, right? This is our gel. Then we put our membrane. Remember the membrane we had up before with our Western blot? We also have one for the Southern blot. And then we have our dry filter paper we put on here. And then we put like some like, I don't know, dry paper towel up here, right? Dry paper towel. And then we put on like some weights. Now what does this do? And we're in a buffer, remember? So this guy is wet down here. What does this do exactly? Well, if you have something wet down here, something dry up here, we're going to have osmotic forces pulling water from something that's of high water concentration to low water concentration. We call it capillary action. All right? Capillary action. And so water molecules will migrate from the wet side to the dry side, right? And as it does that, it will bring our DNA strands onto the membrane. Remember, the membrane is sandwiched in here, right? This is our membrane. And basically what we're left with is a membrane, right? This is our membrane with all our DNA strands, right? So this is basically just the same thing as we did with the Western blot with a different mechanism, all right? And now, okay, what we do is we heat this at 80 degrees Celsius and then we blast it with UV light. And what that basically does is it attaches the DNA to the membrane itself so that it's like covalently attached, right? That's the idea, so that they can't really move. And then we add our DNA probe, sorry? So we add the DNA probe, right? Imagine we have our membrane. Let's say our gene of interest is this green thing in here, right? We add our DNA probe, right? This probe is chilling now, right? We're covered with that probe. Then we wash it, right? We wash it, and all that's left is the probe that remained on the green. And this probe will have a little indicator, right? And that indicator we can see usually with X-ray film detection. And basically what we do here is that we have an X-ray film, right? We blast this with X-ray rays, and then if we have a white spot, that means we had this marker. And I'll show you some pictures of the results. Here they inverted it, right, so that we can see it as black. And we can see in this case, D1 had 3 instances of this sequence, right? And it should make sense why there are 3 different ones, right? Like if you have a digest enzyme, right, and you split this genome into a bunch of different pieces, chances are different parts that contain the gene will have different lengths, right? Because there's a lot of variance in terms of genetic sequences. So if we have multiple instances of this gene across different places on this genome, right, chances are when we do our restriction digest we're going to get 3 different genes, right, of different lengths. For example, if we had, well, let's have this, right? Let's say we have 3 genes here, right? Our digest enzyme, right, might cut it like this, and then like this. And we see we get 3 different lengths, right, that contain the gene. And that's basically what we're seeing here, right? Or like here, for example. And so this basically just lets us see where these genes are. Or sorry, how many of these genes are there. And if they're there, right? That's basically the idea of the southern blot. Very similarly, the northern blot is basically the exact same thing, except we use it with RNA instead. So motivation, this is a very good thing for doing, measuring gene expression. Because if we think about our transcriptome, right, if we have a lot of RNA of a certain gene, that means that gene is being expressed more, right? Whereas before we were looking at if the gene is even there, now we're looking at how much of it is there, right? For example, we can take a skin cell, a skin epithelial cell, right? This is an epithelial cell. And then we have a pancreas cell. I don't know what they look like. I'm assuming they look like something like that. And obviously, pancreas cells secrete insulin, right? And let's say instead of looking at the protein insulin, let's say somehow it's easier to look at the RNA. So let's say, obviously, the the pancreas cell is going to have a lot of insulin RNA that encodes for insulin, whereas the epithelial cell will have none, right? Because it doesn't encode for insulin. Like, it has a gene. The gene is there, right? Remember, we share the same genome, right? We share the same genome, but the gene is only being expressed in our pancreas cell. So obviously here, we don't want to use the southern blot. That wouldn't make sense because we would get a positive result for both. Instead, we want to look at the RNA because this guy will have RNA for insulin, whereas this guy won't. Or maybe he will for some reason, but it will be a lot less, right? And that's the whole idea. Now instead of DNA probes, we use RNA probes, right? And obviously this detects the presence of RNA molecules. And that should make sense. Basically, what we do is replace DNA with RNA. The only difference is that when we prepare our RNA, right, unlike DNA, we have to treat it with formaldehyde because RNA can form secondary structures. If you imagine tRNA, right? tRNA looks something like this, like some three-leaf clover type, whatever. These guys form like H1s here. This is a secondary structure that RNA can form, whereas formaldehyde will basically denature this guy into just one long strand. And so then we can run agarose electrophoresis in the same way, and literally everything else is the exact same. We have our wet filter paper, our membrane, our gel, our dry filter paper, dry paper towel, weight, right? And then we do the same thing. Capillary reaction goes up, and we transfer all of the RNA onto the membrane. Sorry, the gel should be under the membrane. That's my bad. Gel, and then we have our little membrane. This is dry, wet, dry, right? So capillary reaction, and then we're left with a membrane that has all our RNA strands. Remember, we do the same thing. We heat it at 80 degrees. We blast it with UV light. Basically makes the RNA attached to the membrane. Then we do the same thing with this is the RNA probe. We add our RNA probe, and then it'll bind to the RNA sequence, right? And then we'll wash it to remove any extra probe. So we're just left, let's say the probe was for this RNA, right? And then we do the exact same thing with our X-ray film detection, since we have some detector here. And so if you look at some results, these are also inverted in color. But we can see for this EGF is epidermal growth factor, right? We can see that cancer has a lot higher expression than normal cells, right? And imagine we did this with the southern blot instead. We would get positive across the whole thing, right? We wouldn't be able to see a difference. But in gene expression, we can see that cancer cells express this gene a lot more. Obviously, we can do this with the protein as well, but I don't know. If we want to just look at the gene expression itself, maybe the protein, there's something wrong with the protein, maybe it's not stable or anything, right? Then we can use the northern blot to basically see how high the relative abundance of RNA is. And that's basically our three blotting techniques. So if we recap, our western blot was for DNA, for protein, sorry. And remember, we used our primary antibody, secondary antibody. Secondary antibody had our dye or our indicator, and it bound to the primary antibody, and the primary antibody bound to the protein. Remember, so we have our protein, and then we have our primary antibody, and then we have our secondary antibody with our probe. Oh, sorry, not our probe, our dye. And that's basically how we find the Western blot, right? Remember, these proteins are on a gel, right? And on a membrane, right? Remember, we have our proteins on a gel that we ran SDS page with. We transferred onto a membrane, and then we blocked it, and then we added these antibodies. The Southern blot is for DNA. And remember, we ran agarose gel electrophoresis to separate a bunch of DNA strands, transferred it onto a membrane via capillary action, right? And then we added our DNA probes, which has some sort of indicator that we can then use X-ray film detection to see relative abundance. This we can detect either presence of a gene, right, or how many copies of that gene exist in that organism or in that genome. Likewise, the Northern blot is for RNA and does the exact same thing except for RNA, right? And this is good for detecting gene expression. And a big difference between these two is that RNA has to be treated with formaldehyde. Remember, formaldehyde. And this denatures RNA to prevent them from forming secondary structures, whereas DNA doesn't do that. DNA, remember, we treat it with NaOH to break those H bonds between the base pairs to form single-stranded DNA strands. Hope that helped. Hope this makes sense for the blotting techniques. There are more blotting techniques, but these are the three main ones, and you can obviously see why. We have our three parts of the central dogma of, biology or whatever it is, right? We have DNA, RNA, proteins, right? And so obviously this is very, very relevant. Hope that made sense. And yeah.